my name is Olga, as I said, and I'm based in Galway at GMIT, and I'm a postdoc and a senior researcher in data analytics. I'm sure you've seen this abstract here. So first question, what is a stroke? So why do we care about it? So by definition, stroke happens when we have the blood supply to part of the brain is interrupted, okay, or reduced temporarily. So at this time, brain tissue starts to deprive of oxygen and nutrients. And it's a very acute situation. Within minutes, a person will die, most certain. Okay, so we can group them in two categories. So 20% of those strokes can be caused by a rupture or leakage from blood vessel in your brain, and there's a huge 80% of the cases which is due to blood clots, okay? It's so-called ischemic stroke, maybe you heard of that. So I'll be focusing on those 80% because this is the most common one. Okay, so this is a quick visualization for you. So we have, if blood clot is formed in the heart, for instance, but can be formed anywhere, it can travel up the carotid artery, and as soon as it enters your head, it can logit, and this is what is the situation when stroke may happen. Okay, so among risk factors for strokes, there is a so-called atrial fibrillation. I'm sure you've heard about it because before I started to work with this data, I heard as well, but I didn't really know what it was. So it appears to be the most common irregular heartbeat among population. So most people, like I hope all of us, have a regular heartbeat, but if someone has an atrial fibrillation, it is a regular heartbeat which can cause strokes, or to put it precisely, it can cause blood clots to form, which will cause stroke, okay? So I put a little video here, I hope it will work, just to give you quick, or maybe I don't have,
Your heart pumps blood around your entire body, coordinated by an incredible electrical system. Normal heartbeats begin when your SA node fires an electrical impulse. This makes your atria contract, forcing blood into your ventricles. When the impulse reaches your AV node, it pauses before moving through your ventricles. This makes your ventricles contract, forcing blood out of your heart and around your body. If you have atrial fibrillation, the electrical impulses might start in other parts of your atria. These impulses can make your atria quiver or fibrillate. When the impulses reach your ventricles, they will also beat irregularly. Over time, atrial fibrillation can lead to clots forming in your atria, which increases your risk of stroke. Find out more.
Okay. So I hope it gives you a little bit more understanding why this is an important factor and why we are looking into this kind of questions. Okay.
About atrial fibrillation.
Oh no, it's a Jupyter notebook as usual. Okay, so we understand that stroke is a medical emergency, and prompt treatment is crucial. So we need to gain insight into factors which are causing strokes. We know that stroke is a leading cause of disability, and it drains economy. And we know that in Ireland, there are 2,000 people dying as a result of acute ischemic stroke. Okay, so therefore we will be looking into factors causing this. So therefore objectives of this particular project is to investigate risk factors for strokes in a simulated settings. So this data is derived from a simulated study performed by one PhD student in my department. And in a moment I'll show you how it looked like. So to give you some background, using that data, I'm going to apply supervised machine learning classifiers to try to detect stroke, and I will be looking at the different impact variables or features such as a heartbeat, so atrial fibrillation, one of the common ones, but I'll have a few other options, shape of aortic arch, and a variety of clot dimensions, so I'll discuss this in the following slides. So description of simulation system, I'll try to explain this, but this is, I've seen this in real life, how it works. But this is not my system, and I'm not an engineer in medical sciences. So, but basically, as you can see, there is a simulated model here, in the middle. So what they did, they took several images from hospitals, from real people. They pre-selected them, and they selected those which are of interest. So let's say different shapes of aortic arch, which are known to cause strokes or known not to cause strokes. So they wanted to look into data. So for each of those images, so we had in total 4 images. A silicon model was made. Well, first of all, with a hard model with its 3D printing, and then they put silicone around it, and they extract it. So there is a silicone model here, which totally replicates a human blood system around the heart. And we had computers with a software which would simulate different heartbeats. So like normal heartbeat, like normal people have, arterial fibrillation, and also a steady, but steady doesn't really, nobody has it. But this is just to test the system, to know if in case we get something exciting with steady, it means something's going wrong, right? So it can't happen. And and blood clots were made in the lab from cow's blood. So special powder was added, so blood would coagulate, and they would cut it manually and measure it manually and release it manually. And there is a camera here which would record whole process, and it was done more than 1,000 times. So you can imagine what type of project it is. It was a 4 years PhD from someone. So I was happy to get data. So just a quick one here. What libraries basically used, maybe for some of you will be boring, for others not, but so basically pandas, of course, because it's data frames. I used NumPy, matplotlib for visualization purposes, a little bit of Seaborn. For analytical part is scikit-learn. And this new library, CutBoost, I'll talk about it later, but I've never heard about it until 2 months ago, and then I went on a conference in EuroSciPy, and there was an amazing presentation, and I was really impressed, so I decided to try it. Okay, so but I'll discuss it later on. Okay, so this is data. As I said, we have about 11,000 entries, so 11,000 times Clot was released in different combination of variables, and we have 8 features. So among the features, we have a shape of the arch. So next slide will explain better. I'll show you a picture. Depending on what it can be rounded. Like if you imagine heart is going here, it can be like rounded. Most people, 80% of the population would have a rounded one, which is normal. Some people have kind of pointy. It's about 4% of the population, so it goes like that. And the difference would be positioning on branches on aorta, which can all affect, you'll be surprised, but those kind of physical properties of like a vessel will affect probability of having a stroke or not having one. So we are all born with some unique heart system, so, you know, which we don't know. Okay, so this is how it looks like, just a visualization. So you can see, so heart would be here, yeah, and I should say that a blood clot can enter ascending aorta. It may go anywhere here in one of those vessels, okay, or it could go down to descending aorta. So it might not enter any of those, but can go down. And about 80 or 70% of blood clots released, they didn't enter any of those. So there was no stroke, okay? But to be precise, for stroke, only this one and this one, they lead to head. Other ones don't lead to head, it can go somewhere else, for instance, kidneys or something, which is still not nice. If you have a blood clot going into your kidneys, there will be a problem, but at least you are not dead. You might be kind of disabled, but it's not end of the game, right? But so we are particularly interested in those cases when blood clot did go to one of those vessels which lead to the head, okay? So it's kind of a simplification. So arch, just if you go on the previous slide, so aortic arch is this part here, okay? And we say that it's a portion of the main artery that bends between the ascending and descending aorta, okay? So as I said, different people have different shapes. Majority, about 80% have rounded, but some don't. So I say we have type of aortic arch. In my model, I should call them just model 1 to 4. So model 1 is 80% population, therefore you can see that they were particularly interested in that model, and half of the clots were released for that model as well, because it is most of us. Other models had kind of equal number of clots. Okay, so how models were made, I already briefly mentioned earlier, but so there were pre-selected scans from hospitals, from real people. This data is anonymized, we don't know who they are, but this is some people who are really existing. And for each of those, so model 1 through 4, a heart model was made with the help of 3D printing, and after that, silicone model was made. You can see the difference here would be in the shape and the position of those branches here. So sometimes it goes a little bit far apart, and sometimes it's just a bit more pointy shape. Okay, so for branches, we've seen it earlier, we have 5 possibilities, actually 4 branches and 1 descending aorta, so clot can go in any of those. For the purpose of our analysis, because we are interested whether there will be stroke or no stroke, we combine 2 of those which are responsible for leading to the head as we call it stroke, or we call it head. So if every time I say head, it means a stroke, it's the same, it's just going up, or no stroke, okay? So again, so you look here, so those 2 would be going to the head. So yeah, here if you look at the distribution, we see that the majority of clots released went through the descending branch, so there was no stroke. Okay, no stroke, and it didn't go anywhere, it just went down. And because we have, prior to starting this study, there is a whole body of literature suggesting that length of the blood clot or size of the blood clot, including length and diameter, can have an impact on probability of clot going to the head or not going. Okay, so imagine we have a blood clot somewhere formed, it's kind of travels up, depending on how big it is, it's likely, more or less likely to enter head. So we would like to know that. Yeah, so, just to mention here point that for this model they were interested particularly in all kinds of possibilities. So they included extra long clots which are 10 millimeters, so it's 1 centimeter, I can imagine quite big. But I didn't really like the idea of having this skewed data with a long right tail, because from here you get all kinds of problems in the model fitting and performance. So for this data, well, it should be millimeters, of course. For this data, I truncated it, so I have this nice, nice distribution, nearly like a Gaussian distribution for data. Okay, so now yes, we check. So on general, mean size of the blood clot going into any branches is about the same, looks the same, but if we visualize it, we can see, which is very interesting, is that majority of released clots do go through descending branch, and they were also the smallest clots. Okay, so this is important because it will have implication later on after I just introduce you results of the study, you will understand why. So based from the data description, we see that small ones don't go into the head. So here there is a hypothesis, because I've been discussing these results with the experts, with medical engineers, saying why is it? They think that when it's long, it can break in parts and it will still enter your head. If it's short, it kind of goes with the flow due to mechanics, you know. So we're talking about human system, but it's very much kind of mechanical system in a way. There is a flow which will carry it away, and if it's a small one, it will go down, it will be flushed away. If it's a big one, it may break. Okay, so here this is, yeah, kind of suggests the same idea. So the longest clots on average, you see here, right, they do go to the head. So not necessarily, but on average they are the longest ones, and this suggests that the clot length can be a risk factor for strokes, so size of it. So yeah, so I introduced this earlier, so we say that 80% of the clots don't go to the head and only 13% do go. We'll be looking into that. Another parameter which relates to clot is the diameter of the clot. Here is just opposite. So we can see that clots of various diameter can go to the head, but very large diameter doesn't seem to go to the head. So how to understand this? Imagine clot can be thick, so it can't break into 2 parts, it's quite sturdy, so it will just be flushed away. And we suspect that there is a higher degree of interaction between diameter and length, which we just basic plotting here, we can see that there is some kind of linearity, but imagine if clot is long and thick, it's unlikely to break, so it's probably won't go to the head. So we want to explore those questions. Okay, so flow rate, it's a movement of blood through the vessel, tissue, or organ by definition. And types considered, we talked earlier about atrial fibrillation, which is a really recognized risk for strokes. It can, because when blood is, imagine when blood is not constantly pumping, if it's just still for a little bit, blood can coagulate, and this is how a clot can be formed. So it has a higher risk for forming a clot, and therefore a higher risk for getting a stroke. Normal one is all of us, I hope, and steady doesn't exist, it's just a parameter to check the model. Okay, so here we look again in different way with the help some of box plots. We see that longer clots are more likely to go into the head. So if you look here, right, on this part, so atrial fibrillation, RF is atrial fibrillation, and it's kind of slight, hypothesis here is that those which are, so people with atrial fibrillation and longer clots are more likely to get strokes again. So this kind of analysis is like preliminary, but it's very important to get a right hypothesis, so we are testing right things and not jump straight away into some modeling and without understanding the data. Well, this is not very interesting. I think this is just the majority of clots released were for Model 1, and also those, if they do go to the head, it will be from Model 1 as well. So again, from this model, from this picture, we can conclude that atrial fibrillation seems to be a contributing factor for stroke. So I'm stressing this, but I'm kind of leading a hypothesis which we're going to test in a moment. Okay, so I'm going to talk about those two classification algorithms. Logistic regression is a very simple one. I think we should use it just as a baseline to see how it does, because no point of jumping into something complicated without checking your data on something what is readily available and simple. Cut boost is a new one. As I said, I never heard about this before, and this is an algorithm which was it's an open source. It was recently released by Yandex researchers. Yandex, if you don't know, it's a Russian company, very big one. It's about 15 years in the market, and they decided to put their code for everyone to use. And cat boost it's not from cat, it's from category, because it's particularly good with categorical data. When I talk about this, I'll show a little bit more in detail, but just for everyone, if you have some data which is, has many categories, this is particularly good, and they claim that accuracy is very good, which I can, I agree with that because I have the same kind of data. Okay, so first of all, just in case people not really familiar with logistic regression, it's also referred as a logit, or there are different definitions as well, and it's basically a very simple but still often used machine learning classification algorithm that's used to predict the probability of a categorical dependent variable. So we have head, no head, stroke, no stroke. We have a binary. It doesn't have to be binary. It can be more than 2 classes, can be 5, as you like. But you have to realize if you fit into more classes, you have to have more data for support. And I tried for this data to fit more classes just to know precisely where it goes, but it would crash. You need really a lot more data for multi-class classification. And so we have to make some assumption that, yes, so I said we need binary data, which is obvious if we are talking about classification. This factor level 1 of the dependent variable should be outcome of interest. In our case, it is a stroke. Yes, when I say 1, it is a stroke, there is a stroke. It's a positive. So we can't include meaningless variables by saying that, some classification algorithm, you can throw it everything, and then algorithm will sort out whatever is useless, won't just enter model, and that's it. In these cases, it doesn't work that way. So you really have to check that all variables here, all features you are putting in your model, they have some kind of implications. And they can't be multicollinear, which means they have to be really, there should be there shouldn't be any correlation between them, especially no higher degree correlation. This is what means collinearity in this case. And it does require quite large sample size. So in my case, I have 1100. It's not very large, but it's not very small. I think since this data is simulated and it's a lot of manual work, I think it's quite valuable, because this is something you need to imagine. You need to cut every clot and measure it and write down and release it and take pictures, and so it's a lot of work as well. Okay, so traditional steps here, we need to, since we have a categorical data, we need to get dummies, and for this one I just use pandas get dummies method, and so basically we get those variables over there. Okay, and then we combine them into y and x. So y is our response variable, and x is all the rest we are interested in. I used scikit-learn for implementing this, just because it's there, it's simple, it's available. I tested before and it does a good job, so why not? I'm sure probably, well, maybe not all of you know that, but this is a website. It can be used for a variety of problems. it can be simple, efficient tool. You can use it for classification, regression, clustering, all kinds of model preprocessing. So it's very nice library to go and to look at if you haven't done. It's very simple. Okay, so it's from documentation page. And like in traditional approach would be to split your data into train and test. So we set aside a portion of data. In this case, I set aside 30%, which I'm using for testing purposes only, and I train my model on the rest of the data. Yeah, so this is simple as well. I just use train test split method from scikit-learn, and you can see that we have this, for training purposes about 682 clots released, and for testing quite some as well. Model specification, quite standard. I think most of the parameters are left default, except for so you can choose here which algorithm you want to use. And I did try several algorithms just to see if there is any difference. And I think perhaps important part for this data is that I use balanced class weight. So what does it mean? It means that since in my case only 13% of the clots go to the head, data is not balanced, and if you just ignore that fact, you'll get some strange results, which I've seen, and I couldn't understand why it is. But basically, this parameter balanced, it's uses the values of y, which is stroke, having or not having stroke, binary variable, to automatically adjust weights inversely. So in this case, if you have a data and which is unbalanced, check maybe this option, because I found this was very helpful. Model fit, quite easy, just say fit, train, X train, Y train, nothing more than that. And we have a feature importance. Okay, so feature importance in different classification algorithms can mean different or derived in a different way, okay? So since we are talking here about regression or classification, but it's a regression in a way, feature importance is basically regression coefficients here. Okay, so what I did here, I just pulled out regression coefficients from my model. I just transposed them, nothing more, so on a logit scale. So you can't say, you need to do further transformation if you want to be able to say by how much it does change. But, and nothing more than that, I just plotted them. So all features here against this scale, so positive or negative contribution into the likelihood of having a stroke, okay? So the way to interpret this graph is this positive bars mean they contribute positively in the, so they increase likelihood of having a stroke, okay? Negative ones, they decrease likelihood of having a stroke, okay? Does that make sense? So in this case, I think the most important one would be to see that length, yes, it does increase likelihood of having a stroke. So the longer they are, the more likely we have a stroke. Okay. More interesting one is so this model 4. Model 4 I briefly mentioned, but this is a pointy one. So it's when arch is not rounded but really like sharp. You can imagine in terms of dynamics, it may kind of drive this clot through all the way as the blood pass through it, so it does increase likelihood. It's a known fact, and I've been able to demonstrate this here, that it does increase the likelihood. For other part, it seems like a normal flow rate, so majority of people who are healthy have a normal flow rate, it decreases likelihood, so we are less likely to have a stroke. And perhaps not very obvious, but it is the case from those, since those two are negative, steady and normal. Negative baseline is atrial fibrillation. It's not on the graph because it was taken out as a baseline, if you understand what I mean. So from here we can say that since these two are negative, the baseline would be positive, but it's not on the graph. So I'm saying here that atrial fibrillation is likely to increase likelihood of having a stroke. Okay, so I summarize here what I just explained. So again, just remember that this model, it has seems to have an increase in likelihood and stroke and length of the clot seems to increase likelihood. Okay, so if you want to say something about quality of your model, you need to look at variety of tools, including classification report. I just pulled out most interesting parameters out of there, and as you can see here, model is not really good. It's only 50%, so it's like if I just randomly say yes or no, it will be as good, okay? But since we get those other like features, they do make sense. they are according to what we expected, and perhaps more tuning is needed here, but as a baseline model, we are good to go, we good to continue with this model. We look also at precision and recall. I find myself also these two parameters are a little bit confusing. If you don't know, I just pull out here definition. So precision is 10%, okay, so what it does for us, it's basically the ability of the classifier not to label a sample as positive if it is a negative, okay? So negative means no stroke, so we don't say there is a stroke if there is no stroke in this case, so it has a quite low precision. And recall is about 40%. It's a ability of the classifier to find all the positive samples, okay? So all the strokes. So we have 40% ability of labeling something as a 1 when it's a 1, okay? If it's a lot or a little, you will know in the moment when I show you next model and I compare them side by side. It's very interesting, but just remember a little bit that, but I'll show it in the next slides again. And finally, confusion matrix. I hope it's not too confusing because it's simple, but yet can be confusing. So basically what you want to see here, so actual labels are plotted on X and predicted labels on Y. Basically, we are interested in main diagonal. So main diagonal is our true positives, true negatives. So this is what when model does great job, you will have 0 here, and here will be filled, all your samples will be spread here. This is ideal case, never happened, I've never seen it, at least for real data. For simulated, I mean real data in a way that, you know, not simulated by computers. And how bad is other values? So in total we have, for this model, about 293 predictions, which is the trained part of, test part of the model, sorry. True negatives and true positive is a grade. Okay, so this is our ability of model to classify those as a negative when they are negative and classify as positive when they are positive, which is great. But we have false negatives and false positives. So what does it mean? So false positive is not as bad because it's kind of, you can think about it as a precautionary principle. Imagine you say, so you predict positive, you predict a stroke, but there will be no stroke. So a person comes to you and you say, oh, okay, based on this, you are likely to have a stroke. Okay, this is not a great news, but it's better to be careful than, you know, how do we say, than pity, you know, after that, when something happens and you didn't get any warning, then it's bad, okay? So in terms of model, performance is not great, but in terms of problem at hand, I think this is better to have that. And false negative is 23, okay, I think it's bad because model is not able to identify those cases as having a stroke and labels them falsely as a negative, okay? Again, this will make more sense in the context in comparison with other model. You will see how it can be, if it's good or bad. Okay, and finally, I did some cross-validation. You can do a lot more. You can do all kinds of post-processing methods on that, which I am not presenting just for purpose of time saving, but we did a cross-validation, so we fitted 10-fold cross-validation just to check if we'll get kind of same type of accuracy, and yes, it slightly goes up, but it's about the same, so we say the average accuracy remain very close to that of the logistic regression, hence we can conclude that model generalizes well, okay? It's not a great accuracy, but it's not just accuracy what matters, as you know. It's a precision and recall what matters, how many false positive, false negatives you have. So just accuracy is a wrong measure, it's just, it should be looked into other parameters as well. Okay, and finally, Roc curve, which is receive operator curve. If you do classification problems, you probably know this. If you don't, you probably won't. But basically what it does, it plots false true positive rate, which is also called sensitivity against false positive, which is called specificity. Yeah, always happens. And so it's not really good, you can see. So ideally, we want this blue curve to be here in the right corner. Right top, you see? The higher it is in that corner, the better it is. Again, I've only seen perfect pictures on this curve when it's a textbook example somewhere. I've never seen it like that. But so just to give you an impression. And so to conclude, for this part of the, for this classification algorithm, it's not very accurate. It's vulnerable to overfitting. It doesn't perform very well when you have correlated variables. So what can we do to improve it? Or can we try some regularization techniques on this? Or maybe this algorithm doesn't fit our data at all, but this is still useful to start with as a baseline so we can compare all following one based on this one. And it's not able to handle a large number of categorical features. Well, I would say 6 is, in my case, is not large, but because data set is not so big, so it will consume a lot of degrees of freedom in the long term if you all recode them. So after this, I decided, okay, so I tried support vector machine. Result was very bad. I tried a few other things, they were not really good, and the one which really was a little bit, different is this cat boost, as I mentioned earlier. So they say it's a fast, scalable, high-performance, open-source gradient boosting on decision trees. So it's a gradient boosting algorithm. So it's completely different. Now we're talking about trees, okay? It's a library which can be used for ranking, classification, regression, all kinds of tasks, and it can be on CPU and GPU both. And it was developed by Yandex researchers in Moscow. And it can be used for all kinds of problems, and the code is there. So I have nothing to do with them, but I tried, and they're great. Like, they have notebooks explaining, so it's really nice to look at, maybe, if you have kind of data. Okay, so the Git page, you can see they're quite active. So every time I look at it, it's like within hours they're committing something, and there are like 3,000 or 4,000 stars. So it's growing. So features, again, it has it's specifically good for categorical feature support. It can be used on GPU, which, for me it's not necessary, data is small, but if in your case you have large data with categorical variables, you can have a look. It has improved accuracy, so they claim, and I'll show you in a moment. It's very fast, it has a friendly user interface, so if you run it in notebook, you will have all kinds of visualization interactive, which will show you train laws and all kinds of implemented kind of diagnostic tools already. Okay, so basic setup looks the same, really. I just import data, recode it as before. I truncate this 10 millimeters data because I don't like to have this long-scaled, right tail. This is a little bit different because since it's specifically good for categorical variables, I don't need to recode manually dummies anymore. All I have to do is just to say what features are categorical, which I do here. I just select from my data array of positioning of the features which are categorical, and I supply it in the model, and I say, this is it. There is no more work included for you to recode, to omit something, to whatever. So data looks like that. In this data set, I didn't fit train test, but I also fit train test validation set, so it's a three-way splitting, which in some cases can be better. I don't know. There are advantages and disadvantages of both. Of course, I consume now more data on the validation set as well, but it's still nice to try. Okay, so splitting is quite similar. I still use train test split from scikit-learn, and this is my data set now. So I have slightly less data for training, but for testing is even about the same or even more. Okay. For model specification, since it is a gradient boosting, or it is a model based on trees, you have different parameters, right? So you might be familiar, might be not. So iterations, it's a number of trees we fit. So every time, every model run is a tree we are going to fit, and it's based on the feature. It will split as a having stroke probability or not having a stroke. So I have about 100, not so many, but if I increase it, accuracy would be just the same. Doesn't make sense running it for nothing. Learning rate is how fast you descend along the gradient. So it's a gradient descent. It's based on the gradient descent algorithm. So the lower learning rate means you go slower. Okay. Similar to the previous is that we use this class weight options just to say that we don't have a balanced data. Again, I do just the same because I want to keep all the rest is the same. So I fit this model. Fit is very easy as well. You just say fit as before, you say train, and then you supply a vector of this categorical feature. So I just have to say which one are categorical, so they should be treated in a different way. As I said, you do get all kinds of visualization, nice visualization kind of output, which I set false here because it didn't do well on notebook in the presentation mode. But if you explore this, you will get all nice guard, like a Plotly, you can pull, you can see what happens, and it's really nice. And amazingly, test accuracy is 87%. So yes, they claim it's accurate, it is accurate. Remember, logit was 50%, so 87 is really cool. So it looks, but is this much better than logit? Well, I'll try to show you. I'll skip this one for, it's just an extra step. Before I start comparing, I just say something about feature importance. Feature importance in gradient boosting or in tree-based models is mean different thing, right? So here it is basically how often a particular feature was used for splitting a tree, okay? And I suppose the main message here is that these two guys here, length and diameter, seems to be used really often for splitting a tree, which suggests that those two can be very important. You remember, it's just the same as in logit. Logit also suggested that they were important. Okay, so just remember, they seem to be of highest importance. And the nice thing about this algorithm, which is not the case with logit, but it is a case with all kinds of gradient or like boosting trees, is that you can say something about interaction among features. So you just pull it out with a get feature importance, and you have interaction between, I just showed top three, but thing to see here is that it seems that there is a highest degree of interaction between first and second, which is diameter and length. So yes, diameter and length have to interact, seem to interact a lot and seem to affect the probability of having a stroke. Again, classification report, test accuracy really nice, but precision and recall are 0, so model is not able to label positive cases. I'm just running ahead of here, but so we say that again, precision is the ability to classify, not to label sample as positive. If it's negative, it's absolutely 0, so it's not able to do that. Its model only can tell you when someone is likely not to have a stroke. So if you come to a doctor and you use this model, it can tell you, oh, you're okay, you won't have a stroke. But if you have a stroke, the model won't tell you that. Okay, so you can see it again from confusion metrics. So this 0 is good. This 38 is not good, but in logit it was 24, so not far beyond. Not really. So it's really good in saying you won't have a stroke. Model does great. So this is just to illustrate that if you just look at the accuracy of some model, you can be misled. Don't look at accuracy, look at the complex of parameters if you want to say something. Okay, so again, just the same kind of comparison result from that model. As I said already, false positives of 0 are good. But false negative, well, it's not 43, it's 38, but I couldn't update notebook for some reason. It's bad. So it's basically, if there is a risk for stroke, model won't predict it in this case. Also, accuracy is 87%. So ROC looks quite similar to previous one, so I wouldn't see any difference with logit. And finally, comparing two of those, okay? And from here, I would like to get your feedback and maybe some suggestions what to try next, because I do want to get a better output. We say, yes, there is an improved accuracy in cat boost compared to logit, and massively 87% seems really good, isn't it? For this data, it seems good. But, and cat boost is really good in detecting true negatives. So it can say that someone won't have a stroke compared to logit. But it won't say you when you will have a stroke, okay? So I suppose this is always a good idea to have a few algorithms side by side to see advantages, disadvantages, and from here I suppose more algorithms can be tested, or we can spend more time trying to reparameterize our data or our algorithms just to see if we can improve results. And finally, just to conclude, how does it make all sense? Just to pull all knowledge we collected back into, put in place. Clot size, which is diameter and length, seem to be the most important factors, okay? So we've shown that there is a high degree of interaction between the two, okay? And in particular, clot length increases the likelihood, so the longer clot, the more likely to go to head. Our theory is because it can break. And clot diameter decreases, because if it's long and thick, it kind of won't break, so it'll just be flushed in the descending aorta. Normal flow rate decreases the likelihood of having a stroke, which is great news. It means normal people with normal flow rate, are less likely to have a stroke. And arterial fibrillation does indeed increase likelihood of having a stroke, which is in line with all other knowledge. So I suppose main message here, all the models are quite different, and they are not perfect, but they do give us a message which we expected, and we can justify it in terms of knowledge we have already. And so I think it's where to go from here. So we can try different models, we can try different tuning algorithms, we can try different classification algorithms. And finally, I'm finished. If you liked notebook, you can go here. I just put it there and I'll update it again. And thank you so much for your attention.
Thank you very much.